This section explains why light sheet microscopy is fast and gentle in a single stroke. Every prior microscope floods the whole light cone, but wants only one plane, then discards the rest. Confocal rejects out-of-focus light through a pinhole, serially, point by point. Light sheet inverts the geometry. It illuminates only the plane U image, a thin sheet fired orthogonal to the detection axis, so out-of-focus light is never generated in the first place. Optical sectioning becomes a property of the illumination, not a filter applied after. Two things then fall out for free. The whole plane exposes at once onto a camera, a massive parallelism that captures a whole zebrafish brain in under a second, and the sample sees light only where it is being read, which means minimal photo damage. Speed and gentleness are one move, counted twice. This section explains what a light-emitting sensor is, and why it is the mirror image of a light-driven actuator. Optogenetics splits along a single axis, the direction of the coupling between light and protein. An actuator lets incident light drive a molecular event, light in, action out. A sensor runs that same transduction backwards, molecular event in, photons out. It is a genetically encoded probe whose emission is a function of the variable you want to watch, whether that is calcium, voltage, quinase state, or a binding event. Part 2 collects the read half of the field, the fluorogen-based markers and reporter proteins that turn an invisible cell state into an optical signal. The primitive to carry away is this. A reporter is an actuator with its causal arrow reversed, the same molecular interface, the opposite direction of signal flow. Write and read share the one optical channel. Only direction distinguishes them, and together they close the loop. This section explains how fluorogen-based reporters turn light itself into a binding event. Split a fluorophore into two dark halves, and emission becomes the signal that they have found each other. A classic GFP carries its chromophore matured and sealed inside the barrel, always glowing. Gaudier's family externalizes it instead, a small dark dye, the fluorogen, plus a genetically encoded protein, each one non-fluorescent alone, emitting only as a complex. That single move buys reversibility. The dye exchanges on and off, unbound probe stays dark, background vanishes, and labeling becomes a dynamic equilibrium rather than a permanent stain. The boundary is the cost. You must feed the cell an exogenous dye. That is the price of externalizing the chromophore that GFP builds for itself, for free. This section explains why the sensors and the actuators in this book are one device seen from two sides. A reporter and an actuator are the same transducer read in opposite directions. There is one molecular coupling between light and a protein's conformation, and the two roles differ only in which way the arrow points. The actuator half of the book writes in one direction. Photons lead to a conformational change, and that change leads to a cellular action. Chapter 4 is the read half. A cellular state variable, calcium, voltage, pH, or a kinase's activity, bends the same class of engineered protein, and that bend is emitted as photons. That inversion is the whole organizing move of the volume. Light-emitting sensors and light-driven actuators are one channel run in its two directions. To predict any reporter, ask which internal variable is being made observable and through what conformational switch it is coupled. This section explains how light becomes the on-off switch for a signaling protein and why that one trick generalizes across many different photosensory parts. Fuse a signaling protein to a photosensory domain and light becomes its switch. The beam supplies where the activity happens, and the timing of the beam supplies when. That is the whole primitive spatial control from the shape of the light, temporal control from its onset and offset. Here is the founding instance. In Love 2, the J-alpha helix cages the small GTP ACE RAC1 in the dark. Blue light unfolds that helix, RAC1 is released to bind its effector PAK, and the cell edge ruffles in response. This engineered tool is called photoactivatable RAC1, or P, a RAC1. Every other variant is the same move carried by a different light-driven part. VVD, phytochrome B, and cryptochrome 2 each supply their own conformational change, or their own dimerization change, but the logic is identical. So you can predict the next instance yourself. Any activity you can occlude and then restore through a switchable domain becomes addressable in space and in time. The switchable domain is the only requirement. The chapter's own contribution sharpens the specificity. It re-breaks a symmetric homodimer into a charge, complementary heterodimer called the magnets pair. Because the two halves are charge complementary, light now drives one specific A to B pairing, instead of the three pairings a symmetric dimer would allow. This section explains optogenetic control of reactive oxygen generation, a destructive kind of light actuator. Fuse a light-absorbing protein to your target, illuminate it, and the target dies wherever the light lands. This is Kali, the destructive dual of the book's other actuators. It is light that subtracts function rather than adding it. The mechanism collapses to one number. A photosensitizer converts each absorbed photon into singlet oxygen, and singlet oxygen expires within nanometers of its birth. So the damage radius is fixed by the lifetime of the reactive oxygen species, not by the genome. Specificity is inherited from wherever you fuse the chromophore and sharpened by wherever you aim the beam. What you have is a knockout addressed in space and in time by a photon. Loss of function becomes an optical operator, and its resolution is the diffusion length of an excited oxygen. This section explains how optogenetic tools turn a molecular reaction into a quantity you can hold at a chosen value. Two wavelengths of light turn a protein interaction into a knob you can hold at any value along its range, going beyond a simple on-off switch. In the Phi B and PIF system, red light drives the pair together and far. Red light drives them apart. Because the two channels are independent, the light dose sets the fraction of the pair that is dimerized and clamps it there. That clamp is the whole point of the word quantitative. Ordinary perturbation pokes a system and watches what happens. The move in this chapter is to recast perturbation as control. You fix the input at a chosen set point and you read the system's transfer function off the output. Here is the generative pattern for every tool in the book. Light buys you what chemistry cannot: graded, reversible, spatially addressable command over one molecular reaction. This section explains how a kinase becomes a quantitatively controllable actuator once you have measured its transfer function. A kinase is a knob you can only push qualitatively until you know its transfer function. The move in this chapter is to fuse ACT, to cry to, so that light drags it to the plasma membrane where it gets phosphorylated and becomes active, while dark releases it to the cytosol where it dephosphorylates. Location is the activity state, and light sets location. That clean map lets you fit a 4-state mass action model, 2 compartments crossed with 2 phosphostates with light as a 0 or 1 input. Once fitted, the model runs forward as activity expressed as a function of the light signal over time. Activity becomes a predictable function of the illumination waveform. The primitive here is system identification. Measure the mapping from input to output, invert it, and any desired activation pattern becomes programmable. Optogenetics supplies the actuator. The model supplies the transfer function that makes it quantitative.